Hi, and welcome to Chem 200A. Today we're going to do experiment number 5, spectrophotometric analysis of Allura red in Kool-Aid. So this is the setup that we're going to use here in experiment 5, our Kool-Aid experiment. Let's go through some of the equipment that we will be using. We have two graduated cylinders over here. One is a 10 milliliter graduated cylinder, the other is a 50 milliliter graduated cylinder. We will use the graduated cylinders to measure out the appropriate volume of the stock Kool-Aid solution. Our stock Kool-Aid solution is here in this bottle, and we'll measure out the volume in milliliters of the stock solution that we need, and we'll add them to one of these beakers that are over here. In addition to the stock Kool-Aid solution that gets added to the beaker, we will also use again our graduated cylinders to measure out the volume of deionized water that we need to also add to our Kool-Aid solutions. Once we have both the water and the stock solution of Kool-Aid in the beaker, we will give the beaker a good mixing here, and that should mix our Kool-Aid solution really well. We will then use the pipettes, and we'll use these pipettes to transfer our made solutions, both our standard solution and our unknown solution, into our special spectrophotometer test tubes, which are in this test tube rack and come out of this box here. And again, we'll fill those test tubes about 3/4 of the way full. And 3/4 of the way full should be very good for us to use here in our spectrophotometer to get our absorbance values. Now, once we kind of give our solution a mix, we take our pipette and transfer it to our test tubes. Before we're ready to actually put it into our compartment here in our spectrophotometer, what we want to make sure we do is we use this guy over here, and these are what are known as chem wipes. And chem wipes are used to wipe off any type of fingerprints, dirt that may be on the test tube. Again, any of those things, dirt, fingerprints, will interrupt the ability of light to transfer through our solution once it's in the sample compartment. So we always, right before we put it into the sample compartment, want to make sure that we wipe it off with a chem wipe, which again are these guys over here. And thus we can put it into our sample compartment and get our absorbance values. Now, before we actually start taking absorbance values, we do want to calibrate our spectrophotometer, and we want to follow the calibration procedure in the actual manual. The procedure really does call for using a test tube full of deionized water, putting it into our sample compartment, and hitting our calibration button, along with a few other steps that you would obviously want to follow before you take absorbance readings using a spectrophotometer. So you always want to make sure that it is calibrated before you begin this experiment. So let's take a look at the different parts of a spectrophotometer. First off, here is our display. Over here is our transmittance absorbance selector. For our experiment here, we're going to be using absorbance, so we do want it pushed down to absorbance. This is our 100% transmittance over 0 absorbance button. We use that button in the process of calibrating our spectrophotometer. Over here, this is our wavelength selector. We can dial in our wavelength to what we're looking for, and it is in nanometers. And way over here is our sample compartment. And in our sample compartment, that is where we put our test tube that has our samples. Open the lid, put it in, close the lid, and we could get our absorbance reading coming out here in our display. So let's take a little closer look at the spectrophotometer, including the compartment where we put our test tube and the actual test tube itself. Now, this test tube over here is actually a special test tube that is used specifically for this spectrophotometer. What makes it a little special is, as you can see, it does have this marking here, and that marking is really important when we put it into the compartment in our spectrophotometer. We will find a matching marking on the spectrophotometer, and when we put it in, we do want to make sure that our test tube and that line lines up with the spectrophotometer line. We then want to gently put it down all the way as far as it will go down in the compartment. We then want to close the lid, and then we can take our absorbance readings. Now, when we fill our test tube to put into our spectrophotometer, we just need to fill it about 3/4 of the way full with our sample, just tall enough in the test tube that the light will actually pass through our sample and not miss our sample. Again, if we put too little in there, the light will go past this empty spot where there is no sample. So we just want to make sure that we fill the test tube up high enough that the light that is being shined in this compartment here will actually pass through our solution. So now that we've seen a spectrophotometer, let's talk about what's happening inside the spectrophotometer when we actually run our samples. In a spectrophotometer, we have a light source, and this light source is shooting light at a specific wavelength into our sample. And as that light goes into our sample and through our sample, it will reach eventually the detector at the other side. Now it's important, as we talked about, that our sample in our test tube is filled to the proper volume. That way when the light comes through, it will actually go through the actual sample itself that we're testing and not an empty spot in the actual test tube. Now there's two types of light that we see when we use a machine such as this. The light source and the light coming from the light source to our actual sample is what's referred to as the incident light. And the light that goes from our sample all the way to our detector on the other side there is what is referred to as the transmitted light. And sometimes we look at what is sometimes referred to as transmittance of light when we're doing a spectrophotometer type experiment. And that is the ratio of transmitted light, the light that's going from our sample to our detector, over the amount of light that's the incident light, which is going from our light source to our sample. And that's what's referred to as transmittance. The absorbance of a sample, which is really what we're looking at in this experiment, is equal to minus the log of the transmittance. And the absorbance of light is based on the actual color of our sample. So when we have very high absorbance, that means that a lot of the light is getting trapped and absorbed in our sample. And that usually is a result of our sample being darker in color, as opposed to when we have a lighter or lower absorbance, that usually results in a solution that is lighter in color. That means not as much light is being absorbed by the solution, more is reaching the detector, as opposed to a darker colored solution where more of the light is being absorbed and less of it is reaching the actual detector. So there is a relationship between the color that we actually see in the solution and the actual wavelength that we set the spectrophotometer to. Now, for example, in this particular solution, we are looking at a color that is red, and you may say to yourself, well, I must set the spectrophotometer to a wavelength that's in the red part of the spectrum. And actually, what we see in terms of color is the complementary color to the wavelength that's being absorbed. So if we look at our color wheel here, for example, these are our different colors, our RGB as we come around the circle, and the wavelengths that they come about, those different colors in the visible part of the spectrum. So, for example, if we had a blue solution, what that means is because the solution appears blue to us, the spectrophotometer will actually be absorbing the wavelength that's complementary to blue, which in this case would be orange, between say 590 and 630. So an important relationship between the color that we actually observe in the sample of solution and the wavelength that's being absorbed is basically complementary to each other. So again, if we see a purple color, the wavelength of light that's being absorbed will be in the yellow part of the spectrum. If we see a green color solution, it will absorb a wavelength in the red part of the solution. So that may answer a question as to why in this particular experiment our solution is red, but when we set the wavelength on the spectrophotometer, it may not actually fall in the red area of the spectrum in terms of wavelength. It actually will fall in a different part of the spectrum. Let's talk about the relationship that we see between absorbance and concentration. That's given to us by Beer-Lambert law. Beer-Lambert law is over here. That is A is equal to ABC. Let's talk a little bit about the different values that we see in this formula. Capital A is the absorbance, and that's the absorbance values that we get through the spectrophotometer every time we put our sample into it in the compartment and get that reading that we get on the screen. The lowercase a is the molar absorptivity value, our constant, and that's really the ability of the solution to absorb light at a given wavelength. The B is the path length, and the path length is really the distance that the light has to travel through our solution to reach the detector. And C here in our equation is concentration, and that is the concentration of the solution, usually in molarity, which is moles per liter. Now remember that as we talked about earlier, the absorbance of a solution is related to the actual color. So the darker the solution in terms of color, the more light should be absorbed, and we should see a much higher absorbance value. While if we have a solution that's lighter in color, that means more light can actually transfer through the solution and reach the detector. And in terms of absorbance, less is being absorbed by the solution, and we have a much lower value in terms of absorbances. Another important aspect of setting up your spectrophotometer correctly is to choose the maximum wavelength to set the spectrophotometer to. What that means is that's the maximum wavelength for absorption for your particular solution you're dealing with. One way that you can do this is to take your solution and test the absorbance values at different wavelengths. And that's what we see here in this graph. If we look on the x-axis, we have all these different wavelengths. And if we look on the y, we have all these different absorbance values for our same solution. So we would be testing our same solution at different wavelengths. And we can even make a graph like we did here. And we want to look for the maximum wavelength for our particular solution in terms of absorbance. And that can be found at the very top of the peak, right around here in this example solution here. And that's where we would want to set our spectrophotometer to in terms of wavelength. So we would find the maximum absorbance, come down and figure out what that wavelength is, and we would set our wavelength on our spectrophotometer to that value. And that's really important why we would want to set our wavelength to that maximum absorbance. We would want to set it to that maximum absorbance because when we do and we run all of our samples should fall within an absorbance value reading that will actually give us an absorbance value reading. Now let's say, for example, we set the wavelength way over here on the fringe to about 600 nanometers in this particular example. At 600 nanometers, our maximum absorbance is way down here, which means when we go to run our samples, a lot of samples will be above that value for maximum absorbance. And what that's going to essentially give us is a lot of values that are going to be kind of errors. We're not going to be able to actually get absorbance values. So that's why we definitely want to move our wavelength over here to that area where we have maximum absorbance for our particular solution. That way we don't run into any problems when we put our samples into the spectrophotometer that we do not get any readings. We should definitely get all the readings that we need to complete this experiment. This is actually the part of the experiment that is being done by the spectrophotometer master, where they're checking all of the wavelengths to try to figure out what is the optimal wavelength to perform this experiment for our Kool-Aid solution. Let's talk about what you're going to be doing in this experiment. You're going to be making a set of standard Kool-Aid solutions, and those standard solutions, you will know the amount of Kool-Aid that's in there in milliliters. You're going to take those solutions and then run the absorbance values on a spectrophotometer. From those values, both the absorbance values and the milliliters of Kool-Aid in each of those solutions, you'll be able to construct a standard curve such as this one here, where on the y-axis we have the absorbance values. On the x-axis we have basically the milliliters of Kool-Aid in that solution. You'll do a best fit line through all of those absorbance points. So each of these points here represents an absorbance value and a volume of Kool-Aid in each of the individual solutions. When you do a best fit line, you want to draw the best straight line you can through most of the points or near most of the points. You do not want to do a connect the dot type line where you connect each of the dots. Again, it's the best straight line that you could draw based on y equals mx plus b, which is the formula for a straight line, to get a relationship between all the points that you're graphing. And you are also then take your unknown solution, which contains an unknown amount of Kool-Aid. You then can take the absorbance value of your unknown. So for example, if the absorbance value was here, you could then draw a straight line to your line in your standard curve and then go straight down. And when you do so, that should give you the amount of Kool-Aid in milliliters that's in your unknown solution. So we're going to use a set of standard solutions to generate a standard curve such as the one we see here on the screen. You will then use the absorbance value of your unknown and this standard curve to try to figure out how much Kool-Aid in milliliters are in the unknown solution. Part 1 of the experiment, the part where we make the 7 standard solutions, the 1 unknown solution, and the spectrometer masters part to figure out the optimal wavelength to run this experiment. One more time, the equipment used in this experiment. The making of the solutions using a graduated cylinder to add the stock Kool-Aid solution and water to a beaker and given in a mix. The 7 beakers of standard solution and the 1 beaker of unknown solution. After the solutions have been made in the beaker, the 7 solutions and the unknown solution in test tubes. Setting the wavelength. Putting our blank solution into the spectrophotometer. And calibrating the spectrophotometer. The spectrometer masters part for 440 nanometers record the absorbance. For 460 nanometers, record the absorbance. For 480 nanometers, record the absorbance. For 500 nanometers, record the absorbance. For 520 nanometers, record the absorbance. For 540 nanometers, record the absorbance. For 560 nanometers, record the absorbance. Here's a graph of absorbance versus wavelength for what we just did. We can see from the graph that the maximum wavelength is at 500 nanometers. We will set the wavelength to 500 nanometers for this part 2 of this experiment. Part 2 of the experiment, where we run our 7 standard solutions and our 1 unknown solution in the spectrophotometer to get the absorbance values of all the solutions. Absorbance values of our 7 standards and unknown. Record the absorbance value of solution number 1. Record the absorbance value for solution number 2. Record the absorbance value for solution number 3. Record the absorbance value for solution number 4. Record the absorbance value for solution number 5. Record the absorbance value for solution number 6. Record the absorbance value for solution number 7. And now our unknown solution. Record the absorbance value for the unknown solution. All these were recorded at 500 nanometers in terms of the wavelength. Okay, make sure that you complete the rest of the lab, including the post-lab questions. Once again, thank you to Shane for running this experiment.